Right, here's sign sheet. Sorry, this is usually all set up for me ahead of time. I guess somebody restarted the computer. Every time it restarts, I have to reload everything. I did not expect that. I just need to get Zoom. Lectures, this is chapter 18. My husband's like, apparently trick or treating already started. I'm like, when do you think the little kids start, dude? They start when it's light out. Yeah, really. Yeah, I'm trying, man. Believe me. We're gonna do this. We're gonna do the outline version of this chapter. If anybody's really interested in the details, please feel free to go watch one of the recordings. They will have more details than what we're going to cover today, because I'm sure we're all in agreement here that let's GTFO, right? Uh-huh. All right. I promise I'm doing this as fast as I can. Okay, Zoom. Zoom. She signed it for me. You want to sign it? What? I need to tell. I think this gave you last time. All right. So chapter 18 is talking about diagnostic tests. These are going to fall into 3 different categories. These 3 categories are going to be phenotypic. I'm putting it up on the screen, I promise I am. Phenotypic, genotypic, and immunological. Screen start. I just had to get this to start first. There it is. Okay. All righty, for real this time. All right, so those are the ones that we're looking at. Phenotypic, immunologic, these are the orders we're going to go in, and then genotypic, okay? Phenotypic is basically if it doesn't fall into immunologic or genotypic. This isn't like rocket science here, okay? So if it has to deal with an immunological response, if we're recognizing antigen one way or another, that's immunologic. If we're looking at a DNA sequence matching up, that's genotypic. So that's going to talk about PCR. That's going to talk about those endonuclease cuts and things like that. If it's DNA, it's genotypic. If it's immunologic, it might be antibody or T cell, but it's going to be antigen based. Phenotypic is pretty much everything else. So here we're talking about all those biochemical tests that we're learning in lab and that we're going to get our results for today. That's phenotypic. We're looking at, did they express the enzyme or not? They might have genes from mama side that says make the enzyme and genes from dada side that says don't make the enzyme, but are they making the enzyme? Yeah, phenotypic, okay? So we're not looking at the genetics of it. We're looking at what they're actually expressing. For us, when we're looking at the results, we're going to see that in the form of color changes typically, right? So think of it that way. This also refers to point of care diagnostics, where you're going to be at the bedside of your patient doing finger prick analysis. They have machines now that you have little cards that you can insert into the machine and do the blood test right there at the bedside. Point of care has made a drastic improvement in the speed of the care that you can give your patients, especially patients that are like non-emergent, non-urgent care. Like if they're there complaining at the ER because they have a little bit of a fever, you can do a blood test at bedside to see if there's anything metabolically wrong with them. You don't have to wait on the lab to run the CMP or the BMP or whatever it is. All right, so this is our little chart breakdown of our explanation of phenotypic, immunologic, and genotypic. If you're getting a patient who has, you think, meningitis, and you get their CSF sample and you do a gram stain on it right away, that is a phenotypic test, because that's part of just that face value. Do they have or do they not have these traits? You're not doing anything with antibody or antigen. You're not doing anything with their genetics. Phenotypic, okay? We should be adult enough and work in a hospital or been to a hospital or seen a hospital or a vet clinic or whatever. So you know where we get our tests from and how to take them. I would hope that you know that when you are at your patient's bedside, you should be opening a brand new swab tube if you're getting a sample. If it has already been cracked open, I don't care if you just cracked it open in the test room right next to you, the patient room right next to you. You should be cracking open a new one at your patient's bedside every time. You should be collecting your sample and then labeling the thing all right in front of your patient so they can see. If you're dealing with animals, of course, they're not going to be having that issue, but if it's a person, you'd better be doing that. Other than that, collecting the specimens and everything tend to be pretty much the same, whether we're talking about animals or people. Fecal, urine, blood, saliva, all of these things we can do testing on. You might be ordering your tests on paper. This is going to actually be more common in rural clinics and smaller vet offices. Whereas if you're looking at larger vet offices or looking at places like Mercy or Integris or Norman Regional or OU, then you're going to be looking at Epic probably, or something similar to Epic online, where the doctor is going to have already ordered the tests for you as nurses or as techs, and you're just collecting the sample. But I hope that whenever you guys get into the position where you are collecting your blood samples for your patients, that when you print off those stickers from Epic and it says CBC, that you at least know what a CBC is, what it's going to be looking at, all the possible results that you could possibly see in a CBC or a CMP or a BMP, and know the difference between the CMP and the BMP and all of those, and what color tubes go with that without having to always reference something. Epic gives you the stickers with the test name on it and even with the tube color on it. But we should not always just rely on that to be all the information we need, because if your patient is being ordered a CMP to see if they're having kidney failure, you better know why your doctor's ordering that, because that doctor is not always going to be there to talk with the patient. The doctor's not always going to be there to talk with the patient's family, who's going to be more irate than the patient probably, and all of that. So keep that in mind. Epic is great, but don't rely on its knowledge too much. Rely on your own. So, because you will be the experts that they're expecting to hear from more than the doctor, more likely than not. Yes, and this applies in vet offices too, just for those of you who are going to vet, because the vet techs actually interact more with the pet owners than the vets do. And this is the damn truth of it all too. So we like to think that the vets vets do interact more with the pet owners than doctors do with patients, typically. But having said that, the vet techs, the nurses, you guys are the face-to-face people, and you'll be the ones that people are often relying on for comfort and more explanation than what the doctor has time to give. So rely on your expertise, rely on your experience, and don't be that person. They're like, oh, what's a CMP? Why'd they order that? And you'd be like, you know what? I don't know. I don't know. I'm drawing the tube, but yeah, don't be that person. We learned about selective and differential media. If you think that your patient might have staph, your doctor might order for, or you might even, depending on what kind of clinic you're working in, decide to order a specific like selective media to look for staph. Because if you're taking swabs of somebody's skin, they're going to have more than just your target bacteria on their skin. This way, if you think it's staph, go ahead and just select for it ahead of time. If it overgrows with it, you know that patient had a staph infection. It shouldn't be overgrowing with that, if it was just a little bit there. So that's one way you can like get ahead of that game. If you're suspecting that it's something, seeing if it grows on the media that it should grow on. Also, same thing with differential media. If you think that it's something that can ferment and that helps you steer you in the right diagnostic direction, you might choose a media that you're going to look at fermentation. Cool. Dichotomous keys are something that we will be using in our lab at the very end of lab, once we get closer to determining what our organisms are with our unknown. Ours will be set up differently than this one. Ours will be set up as gram positive or gram negative. And then based on that, you will follow, did it ferment this? When it fermented this, did it also make gas? If it did it also use urea? and all these things, and you'll follow your little pathways along to determine what your organism is. The same thing as what this is looking at. So that's called a dichotomous key. Di for two, because there's always two choices for every point on the path. There's not ever going to be more than two. All right, biochemical testing is what we're doing in lab right now. These are all phenotypic tests. Does the organism ferment or not? Does it ferment lactose or glucose or glycerol or sucrose like our organisms do? Can they break down urea? Can they use citrate? Whatever it is, those are all our biochemical pathways that we're analyzing in our bacteria to determine positive or negative on this test. That's a phenotypic trait. It doesn't make the product. We have these cards available to us, these API systems. And I want to point out, this has 50 tests on it. So this might be, you're looking for fermentation of glucose, lactose, glycerol, sucrose. It might have MR on there. It might have VP on there. It might have urea use. It might have the different litmus milk tests, all in these little wells, right? There's 50 tests right here on this card. And this is only looking at different lactobacillus species. So that kind of puts into perspective all the different biochemical tests that are available versus the ones we're actually doing in lab, which is like minuscule compared, which is great. So we're just doing the basics. I'm saving you the trouble. But at the same time, you can just order these cards that have all of those tests. And in one day, you can get your culture, dilute it out with the little reagent, put it in your little wells, incubate it overnight. The next day, you have all of your biochemical results all at once in one place. The results from this, you type it into a system basically, and it gives you, this is E. coli, 99% chance this is E. coli, or 95% chance this is salmonella, whatever. And those cards are useful for that. They're pretty expensive, but if you have a patient that has diarrhea and you know it's bacterial, some tests like this could be useful for you because you don't know which PCR primers to use. You have to know what organism you're looking for to do the PCR, remember? So you can't just do PCR on all diarrheas. That's not how that works. So this can set you up for, okay, the card says it's probably E. coli. Now let's do PCR for E. coli and confirm that. So you can narrow it down that way. So that's using your biochemical tests like we're doing in lab, all in one place. We have antimicrobial susceptibility testing as well. Remember how when you guys put your Kirby-Bauer discs down, like your gram-negative Kirby-Bauer plate might have looked different than another table's Kirby-Bauer gram-negative plate, right? So we can use those profiles of how they react to ampicillin and, I don't know, clindamycin, whatever antibiotic that you want to look at. But we can look at some of those profiles between certain bacteria and expect to see certain sizes of ZOIs and stuff, and use that to determine if that's the bacteria you're looking for as well. This is pretty useful for things like strep, Clostridium, and Pseudomonas species, apparently. This is not widely used, but it can help narrow down when you're trying to compare it to some other things, right? It's not the number one pick, but it's still a phenotypic test that's going to give you information. Another one that's very similar is phage typing. Phage typing is using bacteriophages. So these are viruses that infect bacteria, right? And they're going to, you'll lay down your lawn just like, that's why I'm saying it's similar to Kirby-Bauer, okay? Make your Kirby-Bauer lawn, make dots of phages like you would with your discs of antibiotics, right? Let it incubate. And if you have clearings, again, clearings, we can go by that, right? They're called plaques when you have this. But when you're using the phages, they're called plaques. Those clearings, if you have a clearing, it meant that the phage killed the bacteria in that area. Phages can only infect certain species of bacteria. So this is a useful tool for things like salmonella, especially where we have a broad range of different salmonella bacteria. But if you think someone has diarrhea and it's bacterial, you might put it on the plate and give it phage for salmonella, phage for E. coli, and phages for other diarrhea bacteria as well, like Shigella maybe, I don't know. And then incubate it. And if the salmonella phage causes a plaque, then you know that it has to be salmonella because those phages only infect salmonella. So that's how that would work. Very similar to the antibiotic profiling, but just using specificity of phages for bacteria. Okay, so if you have a diarrhea problem and I were to take a stool sample from you and we get a couple colonies of E. coli growing out on your stool sample, right? Imagine that like you did that for anybody in this room. Would we be surprised to see a couple of colonies of E. coli here or there? Whenever, no, probably not, right? Because most people have E. coli in their poop. No big surprise there. However, if it's like almost all E. coli, then you've got a problem, right? We don't need, that shouldn't be like overgrown with E. coli. We shouldn't have that much E. coli in our system. So looking at what you expect in your normal biota versus what you're seeing in your sample can help you predict what infection is going on as well. Because just seeing any E. coli might not be a problem in a stool sample. Any E. coli in your urine sample, it shouldn't be there, right? Any Mycobacterium tuberculosis anywhere in your body is going to be a problem. You shouldn't have any anywhere. So now we can see that difference between talking about our endogenous bacteria versus those 100% pathogenic guys. Obviously, if you're relying on culturing for some of these things, we're talking about like 24-hour delays minimum, because usually you're going to take the culture and then maybe at the fastest be able to get back to your patient about the culture the next day. Again, that's at the fastest. A lot of these things can also, they'll delay them for like a week if it's not an emergency. So that's the drawback with these tests. And it also says many infectious conditions may actually be caused by non-curable organisms. So I don't know, this basically can cause some wrenches be thrown into the mix if you're trying to treat someone's illness, because it's not always as simple as just getting a diagnosis with something and treating it with antibiotics, right? So whatever. But these are our summaries of our phenotypic tests that we just talked about. Now we're moving on to immunological. This is also, if we're talking about serum, using serum, so that's like your blood tests for your immune tests. The serum is the liquid part. If we coagulate your blood, spin down the tube and take the liquid off, it has your antibodies in it, all right? Or it would have your antigen in it if it was in your bloodstream. So serology is immunological testing based originally off of serum, and it deals with specificity between antibody and antigen. This is all theory that we already know, right? We learned this with ELISA. The theory behind all of this is just that antibody-antigen interaction. It's a very specific interaction. So sometimes it's not always antibody, sometimes it's T cell, but still it's immunological based on that antigen specificity. We can now use this same concept to test not just serum, but urine, CSF, tissue, saliva, whatever it is, and we can put that antibody antigen mindset into play with all those kinds of specimens, still falling under that serology / immunology blanket, okay? These concepts all rely on a concept we already learned. You're going to use known antibody to test for antigen. If you're testing to see if your patient has the antigen, you have to use a known antibody to do that. This is literally what we learned about with ELISA. So this should all be review for us. So if you're asking, does the patient have the antigen? That is a direct test. Does the patient have the antibody? Basically, have they ever seen this thing ever? You're looking for the thing indirectly, right? So that's indirect. So the whole idea is getting those antibody antigen interactions, getting either the antibody from the patient and looking for the reaction, or getting the antigen from the patient and getting the reaction. Either way, whether you're testing for that patient having the antibody or the antigen, you have to have the known other side of it to do the testing. So agglutination and precipitation are two ways that we can look for a reaction. Agglutination, we're looking to see if our antibodies are binding onto cells, or if we're looking at precipitation, we're looking for parts like the antigens, so parts of the things, if they're interacting with antibodies. Either way, these things will create clumps that'll form, and those clumps can either be seen in tiny little wells. They'll create like a blanket almost and lay down, and in tubes, you'll actually see like pellets forming. So it's different depending on the volume, and you don't need to know the difference, okay? All you need to know is that if you have precipitation or agglutination tests, they're looking at antibody-antigen interactions between either with precipitation, soluble, so it's not the whole cells, parts, right? And agglutination is whole cells. That's all you need to know, okay? So this is all these slides are talking about, just so you're aware of what you're missing here. Next thing is immunochromatography. This is using that antibody antigen interaction on a laminar flow surface. Laminar flow is like, oh, how I describe it to people. Turn on your kitchen tap so it's not spraying, not all the way, but in that nice, happy, safe zone where you get that nice, like clean little column of water coming out. There's not air flowing through, and it looks like it's almost just sitting there. Like it could be like not even moving. That's laminar flow, okay? So laminar flow is asking for nice, even flow across a surface. So when you go and take your COVID tests at home or pregnancy tests or whatever, we're trying to get that even flow of liquid across that filter paper or whatever. And when it hits that line where your antibody is that sees your HCG, your human chorionic gonadotropin for a pregnancy test, or it sees the COVID spike protein, that antibody then goes through making its color change thing happen. And that's how we have those color change things going on. So really, literally, immunochromatography is talking about immuno interactions, antibody antigen, chroma being color, so color change, entography, that's all we're referring to with that moving across that laminar flow piece of paper, like you would in a column if you're doing chromatography, columns. But we're using a little piece of paper. Antibody titer. A titer is just a measure of the concentration of antibody in your blood. We can do this by a variety of methods, but you can actually get a titer from your ELISA test based on how dark your color change was as well. We can do this for diagnosing autoimmune disorders, seeing how many antibodies you have against self, as well as antibodies against past exposures or things that are latent or hiding in your body, like HIV. So serotyping is using antibodies to identify different subspecies of bacteria based on specific antigens they may or may not have. This shouldn't be too much of a shock thinking about the fact that E. coli has like the friendly E. coli in your colon right now versus the E. coli that you get on your lettuce where they call all these recalls of your lettuce or whatever other thing you could eat. That's not the same E. coli as the coli that's in your belly right now. That's the like really bad dysentery, bloody diarrhea causing E. coli. So, but they're all both E. coli. That's a species of bacteria. They're not different species. So those might be called serotypes. And that's going to be judged by if they interact with specific antibodies outside of, antibodies aren't always like going to be only reacting with a whole species, right? Some species might have a different marker than one another. Some species of bacteria, you might be Salmonella enteritidis, but you might be the typhoid forming kind or the general Salmonella kind. You might have flagella and you might not. So we can do antibodies against flagella to see if you're the flagella having kind of guy. And that antibody is for serotyping. Then we will call you your serotype flagella guy, basically. So that's that. Western blots are used for confirmation testing. These are a lot like, let's go to the next slide actually. A lot like, this should look familiar, right? Does this look like the DNA tests? Because it should. So it's a lot like the DNA tests. We're going to run your patient serum out on a gel, or we could run out antibodies on a gel. We can do it either way. But let's say we get a patient that we want to see if they have HIV. We'll take your serum, run it out on our gel. The big bits of protein get stuck, just like the big bits of DNA did. The little ones run faster, just like the little bits of DNA did. Same exact concept, okay? Anyways, you run it out on the gel. It's in the gel. That's not particularly useful for you. So you move it out of the gel onto this little filter paper. Then the filter paper has little bits of protein from all over in your serum. You would have protein like all in your little row, all over from all different proteins in your blood, right? But you're only interested in this band here, that little band there. So how are we going to look at that band? Well, we're going to use antibody, obviously, because that's the whole point of this whole entire immunological section. But what's going to be going on here is you now have this filter paper with specific proteins on it, but the rest of the filter paper where we didn't get the proteins transferred is blank filter paper. If you just pour antibody onto this sucker, antibody is also protein, by the way. So it's just going to stick to the background. How's that useful? So what you have to do first is block it. So you do that literally with milk, guys, okay? It has protein in it. So you put your gel, proteins onto the paper, paper into the milk. Now you put your antibody on there, so it will only bind to that one little bar right there if you have it. But can you see that just antibody binding to your little tiny protein bar? Nah, because it's just an antibody binding to a protein bar. Just like with ELISA, when we had our antibody binding, we had to get a second antibody that had the enzyme to cause the color change. That's exactly how the second part of the Western blot's going to work too. You might have enzyme that cleaves something to cause a color change, or you might have a radioactive molecule, or you might have something fluorescent. It doesn't matter, but something that helps you see it. That's what you're going to do next. So patient sample onto membrane, block it, antibody onto it, secondary antibody to see that antibody that causes a color change or visualization of some kind. That's Western blot. This is super specific, highly sensitive. You will do this to confirm your ELISA if you get, a result that you can't really trust. Okay. We're not going to do these questions. Okay. And I'm not necessarily going to give a whole lot of time to them. So I'm going to run through them, giving your questions and your answers so that we can just be sure we're all on the same page real quick. Okay. What type of media is used to encourage the growth of a specific pathogen? Selective, right? We're selecting for something. Number 2, how are biochemical tests used to identify bacteria? Here, you can go 2 ways. Biochemical tests, we can either look at it as, biochemical tests tell us from color changes, what's happening, what enzyme is being produced, are we fermenting, whatever. Or you can even water it down even further and say they're phenotypic. Those are both acceptable answers, okay? Of course, when you take the exam, you'll have choices. So there's that. How is phage typing used to identify? Okay, so actually number 2 reminds me, now that I get to that, because I had a student emailed me about that, because it was like talking about a biochemical test and like, did this fall under a biochemical test or did it fall under phenotype? Because there were 2 questions that were the exact same. And on one answer, it was biochemical test. And on the other answer, it was phenotype. But it was both, because that test happened to be a biochemical test. And because it's a biochemical test, it's also a phenotypic test, because all biochemical tests are phenotypic. Okay. All right. How is phage typing used to identify unknown pathogens? A phage only infects a certain species. So you can see if it infects your bacteria. If it does, it has to be that species of bacteria. Also, we're looking for plaques, just like we would be looking for zones of inhibition. Oh my God. Okay. My husband's like, why aren't you home yet? It is literally 6:15. Okay. What is serology? It's, looking at the immune reaction between antibody antigen response to diagnose. What is an example of commonly used immunochromatography test? One example would be pregnancy. Another example might be COVID. Yeah, I bet there's people at my front door right now. I don't think I need to hear about it every time. All right. Literally the Ring app notifies me every time. This is not the time I need it to be notifying me during freaking trick-or-treating. So, right. There's more immune tests, of course, because that couldn't be the end, could it? This one has the most, details. So this is immunofluorescence, and we can do this directly or indirectly. But either way, what it's going to do is allow us to look at with actual fluorescence, so glowing color, look at the presence of antibody-antigen interaction. So here we have someone's sample. Maybe it's a swab from the genital area, okay? Or from a genital sore, whatever, but it's syphilis in case you can't read what this thing says. That's why I'm talking about genitals all of a sudden. So syphilis, we do a swab in a sore that's in the genital region, and we put it on a slide, incubate it with antibodies that see syphilis because we think it looks like syphilis. And then we can put it under our microscope, shine some lasers at it, and it fluoresces. And then, hey, that's the picture we took. So these are actual syphilis spirochetes. That's the shape of their cells. They're really like almost like a phone cord coiled up. And these guys are covered in antibody that is fluorescent. So those are the actual like cell shape. So that's a direct testing using fluorescent antibody that detects directly onto that specimen. But another thing you could do is have a slide of, let's say, syphilis antigen, and then put patient sample onto that, see if they have antibodies against that, and then put another secondary antibody on that fluoresces. So that's another way you can look at that interaction. So that's immunofluorescence. We already know about ELISA. I don't need to go over it, do I? Indirect and direct. You guys know the ideas with those. There's pictures and definitions on here if you're interested. You can also go back. Feel free. It's the same exact concept as what is in lab, if that made more sense to you. Next is complement fixation. This is literally going off that concept that you have the classical pathway of complement where your antibodies bind to something and that activates your complement cascade. That's all this is based off of. I don't want to go over these because I'm not going to ask you about the exact steps involved in it. Okay. Just know, all for your purposes, that when we're testing, as far as using complement to test, it's based on that principle of activating the complement cascade with antibody, the classical pathway. In vivo testing, we also mentioned this in the last chapter, doing the tuberculin skin test, that PPD skin test, looking for the T cell response. And it is T cell this time, but it's still your immune response that's in your actual living tissue. So that's called in vivo. All right, so this brings us back to this concept, our favorite of all of our terms of all time, specificity and sensitivity, because they're not confusing at all for anybody. So sensitivity, keep in mind, if we're really sensitive, we're sensitive and we get tipped off by everything. So you're going to be really sensitive and pick up every little positive that could be in the area. You're going to see them all because they're all annoying you. So you're sensitive to everything. So you're really sensitive. You have a low false negative. You are very unlikely to call something as negative. That is not the thing that's annoying me, because you're so sensitive, you're going to pick up on everything that's annoying you, right? Very low likelihood of calling something negative when it's positive. Specificity, you are only going to react to the positives. So that's saying, like, I will only call you positive if you're positive. If you're negative, I'm not going to accidentally call you positive. So they have a low false positive rate because that's basically saying they are less likely to call a negative a positive. Okay, that's the difference. Next is our genotypic tests. These are also, a lot of these, if we're talking about PCR or anything else that amplifies DNA or any sort of nucleic acid, these are called nucleic acid amplification tests. My Tuesday morning class, I remembered to remind them. And so now you guys get the reminder as well. When you guys are doing your personal presentation preps, right? That's going to be due, what is it, like on the Sunday before we start unit 4, right? So that's due. Bare minimum, I need you to have your slides like put together. They don't have to be finalized on that day. I'm not grading shit until you guys do your presentations. But I'm sure you guys have figured out by now, maybe you've heard me talking about it. You present on the day that your chapter is. So if your disease is a skin chapter, guess what? You're going first. So sorry, but that's how that goes. That's why I don't notify you of that ahead of time, because this is meant to feel random. You're meant to choose your diseases randomly, because I don't want everybody choosing diseases on the last day of the presentation and having the whole freaking class present on that last day, right? That's just too much. So we get it spread out that way. Everybody's slides are due on that midnight before we start that whole mess. I guess you guys get till maybe Monday. I don't know. I don't remember how I set it up, but whatever. When your slides are due, they're due. If you still need to make edits, feel free to up until the day that you present, the moment that you present, if you want to. I don't care, right? You will get a list of what day you are presenting specifically with your names and your disease and what day you're presenting next week sometime, okay? So that you are prepared, so you know exactly what day you're presenting. If you're not sure if your disease falls onto skin or neuro or cardio or respiratory or GI or sexual, those are our chapters, okay? So we'll have that list going out. But anyways, the two most missed topics on those presentations are not giving enough detail on the testing or not giving enough detail on the treatments. Then I would say after that would be not enough detail on your actual organism. But when I tell you that you're supposed to tell me like how you diagnose the thing, I am not asking you to tell me they do blood tests, because the whole reason we're going over this right now is to remind you that you need to know which ones of the blood tests, what blood tests do they do? If they routinely, let's say your dude chose COVID and they're routinely saying, well, you can do home tests, you better tell me that you better not be leaving it at home test. You better say that is, immunochromatography home tests. And if they're doing the PCR tests, saying that it's a PCR test is probably fine. But if you're presenting your presentation, and I feel like what, generally speaking, of course, that you guys have an idea of what you're talking about when you put it together, that you did your work for it and you're familiar with the concepts you're talking about, you'll be fine. If I feel like you're reading the slides verbatim because you copied and pasted everything from online, I will ask you questions. And I'm going to be pretty loose about it. Like, I'm not necessarily going to be asking everybody a question, but if I think you ain't done your work, you're going to get a question. So you'll know immediately if I don't think you did your work, pretty much is what I'm saying. I'm not calling on everybody. Okay. All right. But that is a warning. Be sure that you have, it doesn't have to be every single test that ever done on that thing. Give a couple examples that came specific, that relate to what we've learned about, and you'll be fine. The other thing was the treatments. Don't just say they treat it with antibiotics, right? Tell me what antibiotics they use or what antivirals. If it is something that they can't treat, like a cold, right? We don't give antivirals for colds. It's perfectly fine to say, or even test for it, right? You just usually go to the doctor. So if you chose something like that, then just say that, but explain out why that's what they do, please. Okay. So yeah, palliative care is fine. Just, be sure that you've done your research. There aren't tests that people do normally. Because yeah, if you have COVID and your treatment normally is for healthy people, we might just stay at home and push fluids and take Tylenol. But if you're in the hospital, that's not going to be the case. So you might want to have otherwise healthy people do this. People in the hospital might be given this medication. Get what I'm saying? Okay, cool. I just want to make sure that we're clear on that because I know that's coming up too. You might not have thought about it yet, and that is fine. But just so you know, preparing. So nucleic acid, gosh, man, this weather has made my allergies go insane. Nucleic acid amplification is all of the PCR tests. There's all sorts of different kinds. Hybridization is where we're using probes. And that is saying, if you have a sequence in your genomic sequence to make a toxin, we can make a little probe that matches that DNA sequence for that toxin, and then see if it matches up to your patient's sample, DNA-wise. So it's very, very similar to that antibody antigen response thing, that specificity, but now we're looking at actual DNA to DNA sequence matching up. So we're using probes like that. Then we have whole genome sequencing, speaks for itself, right? PCR, we're amplifying DNA. We're making copies of that DNA on purpose, doubling it with each round. And you need to have primers to know what to look for. You can't just say, I don't know what it is, let's PCR it. So you need to look for specific things. There's different kinds of PCR. Real time uses A, T, C, and G, each with a different fluorescent color on them. When they get used up, the A turns yellow, the A turns yellow, the C turns green, the G turns blue, the T turns red. I don't know. And as they do that, the machine that it's running in can actually see those colors coming as it happens. If you're not amplifying, if you're negative for that test, you won't be shooting off colors. If you're positive, you'll be shooting off colors, and we can make sure they're in the right sequence that they should be. So you can get in real time, actually see if your patient's going to be positive before that 3 hours is even up for running that. For reverse transcriptase, that is saying we can get an RNA sample and turn it into DNA and then do regular PCR on it, because DNA is more stable than RNA. So you'd want to do it that way. That means you can test for RNA-based viruses, or you can look for, mRNA that might be present in a cell. Multiplex PCR, we're testing for multiple tests all at once. You can go to the doctor now and get one swab done, and in one PCR cycle, get tested for flu, RSV, and COVID all at once in one PCR, because they have the primers set up so that they're so different that they can look at which one's popping positive in it. Okay. Pan-bacterial is just looking for general groups of bacteria. So you might have genes that signify that this is type of bacteria that is in the gut versus gram-positive bacteria versus whatever. That's generalized grouping. So that might help you narrow down. Don't ask me about RT-LAMP because I don't know what it stands for. Hybridization, moving on. Hybridization using those probes, using a little bit of DNA that matches this sequence, popping it with some fluorescence or something so you can see if it bound or not with a microscope. Here's an example. Fish. Fish is fluorescent in situ hybridization. We can use whole intact cells with little probes attached to fluorescence and look for cells in a biopsy sample, a tissue sample of some kind, and look to see where cells are that have these genetic sequences. We can look for genetic sequences that might match cancer sequences, or for even bacterial infections to see if they're infected inside with certain things or with viruses, whatever it is. So use those probes. So here's an example. This is better than me talking about it. Let's show it. This is fish testing for Staph aureus. So like you would have with MRSA. So fluorescent in situ hybridization. We don't have to break open all of these staph cells. This can work with them intact. So we did a blood culture from somebody. We smeared it on a slide. It was gram positive. Okay, we think this might be staph. So we get our fluorescent probes for staph DNA, mix it, and look at it under the microscope, and the cells that have staph DNA will glow. So it's very similar to what we're talking about with those antibodies, right? Except instead of using antibody antigen, we're looking at DNA to DNA sequence matching. So those probes work very similarly. Or you can just sequence for whole genomes. You can do shotgun sequencing. Why is that doing that? We can do shotgun sequencing and like literally if there's an outbreak and we don't know what it is, just do shotgun sequencing to get an idea of what it might be. Gives you a general idea. And now you can narrow your diagnostic procedures based on what you're seeing. It can also tell you about drug resistance. If you start having issues with drug resistance, you can sequence the whole genome and find out where those genes are at and what type of drug resistance it is and how you might battle against that. You can also create microarrays. This is essentially like that fish principle that we had, right? We had the probes that identified Staph aureus. Now imagine that you have a whole bunch of those probes on these little tiny plates, and there's a dot here for Staph aureus, and a dot here for E. coli, and a dot here for C. diff, and a dot here for whatever and whatever. And there's 365 little dots on your plate, each for something different. And now you take your sick patient and you put their sample on there and incubate it on there, and then let the machine see if there's any binding happening. That's what microarrays are. And you're not just looking for DNA presence of infection, but you can look for mutations that might be present associated with certain kinds of cancer. Your patient has breast cancer, but you don't know which type of mutation might be associated with it. Now we have all these plates that are available for you to use that have different versions of this cancer, DNA-wise on there. You can put your patient's DNA samples that you made, prepared, selected, mix it in with that plate, and then let the machine read out where you have matches. And now you can determine what mutations your patient might have that led to that cancer. So very helpful tool, extremely expensive, of course, but it gives you that idea of how to look at those DNA mutations or different DNA sequences for your infectious agents all in one place. In fact, you could imagine, so this is a picture of how that would work. You can imagine that now you can create a plate. Some of this plate has DNA sequence matchup. Some of that plate has antigen antibody interaction and stuff like that. So you can almost create an entire lab on a chip where you could incubate a patient sample with this chip and get DNA or antibody antigen responses all on that one chip, plug it into a machine that's calibrated to that chip, and it tells you what disease your patient has. You don't need a whole freaking lab anymore. You can just do that right there. Of course, this will be expensive up front, but once this gets automated to a point that it's not as expensive, this is something incredibly useful to places that are in rural areas that don't have labs available or don't have the time. Or it's incredibly useful for undeveloped countries that don't have the same medical resources that we have. This is something that we could donate to them pretty easily and would be easily maintained. So that's lab on a chip. There are other tests that we might do that fall into the other category. These are going to be things like imaging and mass spec. These are not necessarily phenotypic or genotypic or immunologic, okay? So these are the other guys. Mass spec, which you might have heard of, mass spectrometry, or a version of that called MALDI-TOF. I don't remember what MALDI stands for. I'm sure it stands for a thing I knew once and I don't know anymore. But TOF stands for time of flight, and I remembered that. But essentially what is going on is I take your patient sample, I shoot lasers at it, legit, like they're already sounding cool, but it's not cool because it's physics and stuff, okay? But you shoot lasers at it and you create ions that just bounce off from all the molecules that it's made up of. And we're just going to continuously shoot lasers at it until we get all of the ions knocked off that we possibly can. The machine measures the charge of those ions you're knocking off. It measures how much time of flight there is, so how much time they spend in the air once they've been ionized. So mass, charge, time of flight, all of this stuff. And based on that, builds an exact chemical profile of all of the atoms and other submolecules in that sample. and then tells you what it is based on that. So we're looking at something at the absolute molecular makeup. It's just nuts. But imagine you have a patient that OD'd on a drug or is toxic from some sort of drug overdose because of misprescribed medication, but you don't know what that drug is because they're not your regular patient and you don't have their files and they're unconscious. But you know it's a drug. You can do MALDI-TOF on their blood sample and it will tell you what drug it is that they overdosed on. It takes a minute, but it'll do it, okay? You can also do that to identify pathogens, and it will tell you exactly how strongly infected you might be based on how many of those ions you get. You can also use something that we're more familiar with, imaging. Cool, right? I don't think you need to hear any more about that. So take pictures inside or outside, and you can pinpoint where infections might be, and then you don't have to do exploratory surgery. You can now do biopsies or needle aspirations instead of subjecting somebody to surgery. Cool? That's it for chapter 18. Let's go up to the lab, guys. Like I said, a lot of it's stuff that you would know anyways.